Okay, let's carry on with our discussion about DNA. Where are we so far? We've learned about basic structure of DNA. We've learned that DNA itself is a double molecule, has two sides, two molecules that are kind of stuck together. And in a gene, one of those strands contains a recipe ultimately for protein that is manufactured by the cell. That recipe is written in words which consist of 4 letters, A, T, G, and C. And the order of those letters is what spells out the code for the protein. So we also learned that any changes in that sequence can be inherited by children, by progeny of whoever had that DNA. And we also learned that DNA replicates, it doubles itself very easily when a cell divides. And it doubles itself by splitting the 2 halves of the molecule apart and then reconstructing the other molecule. And it can do that very easily because those 2 molecules match one another. A always matches to T, T always matches to A, C to G, G to C. In whatever order is on one side, prescribes the order on the other side, which is how DNA replicates so easily. What is going to become very important for us in talking about forensic science is that the replication of DNA begins with a little short stretch of RNA, another nucleic acid molecule, being laid down to match a few of the bases at the start of the place where the DNA has got to be doubled up. And that little patch of RNA, which defines where the replication starts, is called a primer. And we are going to see how we can actually manipulate the system of doubling the DNA to our purposes, and especially important for us for purposes of establishing forensic evidence. The DNA, all of our DNA is absolutely unique. First of all, you need to realize that your mother has a unique combination of genes, which she inherited. They're a mixture of the genes from her mother and father. Your father has a unique genetic makeup made up of a unique mixture of genes that he inherited from his parents. Likewise, each of us is unique. We are genetically unique. Our whole gene makeup, our genome, is made up of a mixture of genetic information from the father and mother. And half of our chromosomes, for example, are from the father, half of the chromosomes are from the mother. Remembering that inside our cells, in the nucleus, each chromosome occurs in pairs. There is a maternal chromosome and a paternal chromosome for each of the 23 pairs of chromosomes that we have. So what you're going to see is that you inherit information from your mother, you inherit information from your father, but your overall genetic makeup is entirely unique. And that becomes very important because it means that if we can find a way of typing the DNA, if we can look at the DNA in particular ways, we can see its individuality, and we can tie a particular DNA to an individual. DNA analysis provides us with very powerful individual evidence. And it's powerful because we know a lot about the human genome. We know a lot about the genes and the makeup of the chromosomes and everything else. We can put statistical bounds on the likelihood that something we see in the DNA, a sequence or whatever that we see in the DNA, we can say how frequently that occurs in the general population. This is tremendously important because it allows us to define our error, which is not something which is always easy to do in forensic science. So first of all, let me explain. There's one way you could determine somebody's individual DNA makeup, and that would be to get a readout of all of the bases, of all of their DNA bases, and determine the sequence of those AATGDAAA for hundreds and hundreds and hundreds of thousands of bases that make up the meters of DNA that we have in our cells. You could do that, but that would be very, very tedious. It would be very time-consuming and very expensive to determine the whole human genome for an individual. It would probably be even more time-consuming to then compare that to everybody else. So we actually use a shortcut in identifying, typing DNA. And we make use of the fact that we already understand there are long stretches of our DNA which have useful code in them. They code for proteins. Those we call the genes. We're not so much interested in the genes, in actual fact, in DNA typing. We're interested in intervening areas of the DNA, and there is a lot of it. A lot of our DNA does not code for anything. It is DNA that occurs in between genes. And its exact function, it does sometimes have function, but it does not code for protein. Now, as we go through the human genome, we look at all of the chromosomes. On all of the chromosomes, there are places, defined places, where something rather strange happens. And that is that segments of DNA, little bits of sequence, repeat themselves over and over and over and over again. We call these tandem repeats. They're tandem repeats because they're next to one another. So they are hugely variable. It depends which ones you're looking at. We know the exact location of most of them. And in some of them, the tandem repeats are very short. They might only be 4 bases. A, G, C, C, A, G, C, C, A, G, C, C, or something like that. In other of these areas, the repeat sequences are actually very long. They can be 13, 15, 20 bases long, repeating one after the other after the other. What is really important is that the number of times they repeat is highly variable in the human population. For any one place where these repeats occur, you might inherit a chromosome that had 5 repeats from your mother and 20 repeats from your father. You go out into the rest of the population and you can determine how many people have an area with 5 repeats, how many people have area with 20 repeats. What are your chances that you inherited 5 from your mother and 20 from your father? We can work it out. That's really tremendously important. So what we're going to see is, for example, that if we just look at one of these sites on the DNA, maybe we end up, as I say, with 5 from the maternal, 20 from the paternal. So we'd have a little formula, 5 and 20 for that particular site. We can then go and look at the whole population and say, how many people in the population also have 5 and 20? And we can say, okay, it's like 5% of the population. Well, that doesn't give you individual identification, does it? No, it doesn't. That gives you class identification. But remember what I told you, there are many, many, many places in the DNA where repeats take place, where these tandem repeats occur. And they are very different to one another. So at R1, 5, and 20, maybe the repeating sequence is a little short thing, A, G, C, C. We go somewhere else to another chromosome. There may be another area of repeats, and we find that there are repeats where the repeating segment is G, 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 G. Quite different, but similar in that we have the repeats one after the other. And at that site on another chromosome, we find we have 4 and 8. So now we've got 2 sites. We've got a 5 and 20, and we've got another one on another chromosome, it's 4 and 8. Now let's go out into the population. And we'll find out, first of all, how many people have that second combination, like 4 and 8. We can say, okay, that's this percentage of the population. What percentage of the population have them both? What are the chances that you would inherit both in the same? It becomes much more remote. And the more we look at these sites of repeating DNA, the more we put together, the greater and greater and greater our certainty is that the DNA that we're looking at from a suspect sample is the same as the DNA that comes from a standard sample if they share the same distribution of these repeats. So first of all, let's get it clear in our heads. There are areas inside the DNA. They do not code for protein. Instead, we don't actually know what their function is, but when we look at their structure, we see 2 things that are really important. For each site, from the father and from the mother, we have an area in the same place on the chromosome where these repeats occur. But the number of repeats from the father may be different from the number of repeats that on the maternal chromosome. That's the first thing. The second thing, which is equally important, is that those areas of repeating DNA are flanked on either side by specific sequence. There's like a label which we know because we've sequenced the human genome. On either side, there is specific label which always occurs there. So we'll see this in more detail in a minute. We know a lot about a lot of these areas of repeating DNA. So they have become very, very important in typing DNA because they allow us to type DNA very rapidly and using standard techniques, which are now basic to laboratory practice. The really important thing to realize is that these areas vary tremendously widely. The number of repeats vary tremendously widely in the human population. So we can work out the probability that you will inherit one at this one site, you will inherit a particular combination. It becomes very powerful when we start to look at more than one site of repeats. Now, I hope that you've all watched that movie, The Footpath Murders, because I'll explain to you now that the very first time this kind of analysis was used was in that case. It was based on research which was already undergoing, but the research was specially adapted to try and identify DNA from a crime scene and then link it to standard DNA taken from a wide variety of people. It wasn't only suspects, but a wide variety of people, and to compare DNA recovered from the scene of these sex murders and try and see if it could be tied to a particular individual. eventually it was. Along the way, the evidence freed an innocent man. And this provided such tremendously powerful validation of the technique of DNA identification by looking at these tandem repeat areas that it rapidly became standard. The very first type of analysis which was done is called restriction, fragment length, polymorphism. Now that it also looks at an area of the DNA where there are lots of repeats in the DNA. And again, there's tremendous variability, and they do it for a couple of different sites in that instance. There's tremendous variability in the human population. But as we start to combine chromosomes from maternal line and from paternal line, the chances of inheriting a particular combination of lengths of these tandem repeats gets smaller and smaller. So here's how this is commonly called RIFLIP, because nobody wants to sit there saying restriction, fragment length, polymorphism over and over again. But nonetheless, let's just have a look. And I'll explain that, again, every part has a meaning. A restriction fragment, we're going to explain now. A restriction fragment is a section of DNA that we cut out with enzymes, with specific enzymes. And when we use this technique, we cut with the enzymes, and the lengths of the DNA segments varies depending on how many repeats there are inside the fragment. So that's the next thing is really important, is that is the length. We're going to be measuring the length of those DNA fragments. Polymorphism, this refers to the fact that there's a huge amount of variability in the population. So what we see when we perform this technique becomes like a fingerprint almost for the person that we're examining, because they end up with a unique combination of different restriction fragment lengths. I'm sorry, I can't seem to explain it any more clearly than that. If you're lost, just back up and try to listen to me again and see if it makes any more sense. But I'll show you in a series of diagrams what we're talking about. Now, the important thing about restriction fragment length is that on either side of the area where these repeats are occurring, there are patches of known DNA sequence where enzymes can cut, like a pair of scissors. We can cut on either side of this fragment, and we can extract it, and we can examine for it. So here's how these are called restriction enzymes. And it's another important thing for you to know just for general knowledge, because it actually forms the basis of a lot of genetic engineering, is our ability to cut DNA at specific places. And the specific places where we cut is determined by short little sequences of the bases. So, for example, there are many of these enzymes which we can use, which will cut DNA, but they all cut at a specific place. They cut at a unique place. This one here, the enzyme is actually called EchoR1, and it always cuts where it sees this little sequence, AATT. It will cut on both sides of the fragment of the DNA, AATT. It doesn't matter. It will always cut AATT where it sees it. So if I've got a length of DNA here, ignore this one. We'll just look at these two. So I've got a length of DNA, and it has that sequence, AATT, AATT. in it there. And if I take the DNA and I put it together with that enzyme, which, by the way, I can buy, it's commercially available, the enzyme will cut the DNA, but it'll cut it there. It will only cut it there. It will only cut where it sees AATT. So let's just say, for the moment, I've got this person here, and I'm cutting AATT. Let's imagine that in between here, there are some of these repeats, right? And on the one chromosome, on the one length of DNA, which makes up the chromosome, in between this cut and this cut, there are 5 repeats. Now they've got another chromosome, which comes from the other parent, but they've inherited something unique from that other parent. They've inherited 20 repeats in between here. So this enzyme cuts on the one side, on the one chromosome, it cuts out 5, and on the other chromosome, it cuts out 20. Here's our restriction fragment. That's our restriction fragment. It's the fragment of DNA which has been snipped out by these enzymes. On the one chromosome, 5. On the other chromosome, 20 of the repeats. This fragment is going to be much, much shorter than the other fragment. The other fragment is 20. This one is going to be 5. We're going to do something. We're going to perform a technique where we actually look and display that difference between the fragments. Now, one of the significant things about the RIFLIP technique is that these repeats are actually very long. They can be between 15 and 35 bases long, and they can repeat thousands of times. So this, in fact, I said 5 and 20. In actual fact, a RIFLIP fragment would be very, very long. It would be a very big fragment. But the principle is exactly the same. And this had already been realized before, that there were these areas of tandem repeats that could be cut out like this. The idea of using it to actually characterize DNA was one that came when the laboratory was presented with the forensic case and asked to devise a technique whereby DNA from a crime scene could be compared to DNA taken from suspects. So the first thing is that these RIFLIP fragments are very big. The second thing is, because they are very big like this, there's a huge amount of variability in the human population as to how long those fragments are. And the chances of inheriting 2 in a particular combination is already very big. Okay, the chances that, I'm sorry, not big, the chances of inheriting a particular combination are very small. The chances are very big that if you identify DNA by RIFLIP from a crime scene and from a suspect, that those DNA sources are the same. So RIFLIP is a very, very powerful technique, and it's quite surprising given that it was the very first one to be developed. But the chances of inheriting a particular RIFLIP combination are usually 1 in many millions. And if you look at more than one RIFLIP site, which you can do because they occur on different chromosomes all over our genome, then the chances become 1 in billions. And it becomes very powerful evidence of individual character. So here's what a RIFLIP fragment would look like. Again, here they've just given you a 15 base, but remember it can be much more than that. It can be 35 bases. This is the 15 base sequence that they give for this, and this would be the same for this one, same for this one, same for this one, same for this one, etc., etc. And the whole area where these repeats occur is flanked by known DNA sequence which we can cut with a restriction enzyme. So getting this DNA sample, remember this is just one of the chromosomes, one DNA molecule, we can cut it with a restriction enzyme, and we'll end up with this fragment here. And it'll end up a large fragment, usually thousands of bases. And then we do this, obviously we're doing the same thing on the other DNA at the same time. It's just not shown here, but the enzyme will also cut from on the other chromosome, the matching. If this was maternal, it'll cut the matching paternal area out as well. But the paternal may be completely different to the maternal. Again, here we have 5, maybe we've got 20 on the paternal again. So now we need to somehow discern that we have cut those things out. How do we do that? There are various techniques for doing it. The simplest technique is this, is to make use of gel electrophoresis. And what we'll do is we'll take our DNA sample, we'll incubate it with the enzymes. The enzymes will cut it up into pieces. And we will then take those samples of DNA and we'll put them into wells cut into a gel. Now this gel is just like jello. It's like very stiff jello. The gel actually acts like a filter. The gel consists of molecules which are all tangled up together with one another, but it's not solid. They have pores in them. It forms a mesh-like structure that molecules can actually pass through. But these tangled threads, if the molecules are passing through, the tangled threads actually retard the molecules. They don't pass through all that easily. So the very first thing, how do we get them to move? Well, we embed first of all, this is a gel. It's made with a liquid. It's made with a liquid containing salts and things. And we put that gel into a bed that is filled with fluid. And the fluid will conduct electricity, and the electricity will be conducted through the gel as well. DNA, fortunately, DNA is negatively charged. So if I put an electrical field here, if I put a positive electrode here and a negative electrode there, and I switch on the electricity, the DNA will start to move through the gel. But as it moves through the gel, the molecules get retarded by the actual gel matrix. And small DNA molecules are retarded less, so they move faster. They move down faster. The bigger molecules are retarded more. They move more slowly. And as they pass through this gel, the small and the large molecules get separated. And they get separated by size. We can run our gel for a certain length of time, and then stop, and we'll stain for the DNA. We'll stain the DNA with a fluorescent dye, examine it under ultraviolet light, take pictures of it if you want, and we get a measure of the different sizes of DNA fragments that we're looking at. And remember, what we did was we're looking at the difference in size of those restriction fragment lengths. So this is the sort of thing that you will end up seeing. And this is down the side here. This is the marker. This is just a ladder which is made up of different known lengths of DNA so that we can get a good size. And we then run the samples, and different fragments will run at different speeds. So this fragment here is very much larger than that fragment. And we can separate out our restriction fragment lengths. And we can do exactly the same experiment on our known samples from the crime scene with the samples from the suspects. And we can see whose DNA matches whose restriction fragment lengths match. And this is exactly what was done in the footpath murders. They ended up being able to match, eventually, match somebody's DNA to DNA taken from the crime scene. And this is exactly what they were doing. They were digesting the DNA in each case with restriction enzymes. They were running it out on a gel, and they were examining to see where were the same restriction fragment lengths observed. Crime scene and suspect. Most of the suspects, there was none. There was no similarity, but one person had the same, exactly the same restriction fragment length. And the probability of that occurring by chance were infinitesimally small. This is one big advantage of RIFLIP. RIFLIP, you don't have to look at many different RIFLIP sites because they are very big, and the amount of variability in the human population is huge because they are so large. Here's the typical one. Let's see if we can spot who's the guilty party. Here's the crime scene. This would be typical for RIFLIP. Here's the crime scene, and here are the fragment lengths, the restriction fragment lengths there. That's how they've run out. Each of them has been cut out with enzymes from all of the DNA. Each of them has a different size. This one here is large. This one here is small, if this is the positive down here. So here's suspect one. Do they match? Well, one or two of the bands do match, but there are significant differences in the patterning. Remember that we're looking at a combination from both chromosomes. So here's suspect number two, a match, a match, a match, match. They match almost perfectly. Suspect three, there's hardly any match at all. They do match the first band, and they seem to match one down here, but that is all. It is clearly suspect two. And with RIFLIP, when you work out what is the probability that this pairing occurred by chance, it's extremely low. RIFLIP is a very, very powerful technique. RIFLIP, however, has one major disadvantage, and that is you need a lot of DNA. Now, if you are dealing with a semen stain, for example, there's plenty of DNA. And this is the most famous case of all, probably. It used exactly the same techniques as the footpath murder case, but this is the case of President Clinton, and this is the infamous blue dress which Monica Lewinsky wore when they had a sexual assignation, and their dress was stained with a semen stain, which was RIFLIP typed, and DNA from the president was also typed, and it was a clear match. It matched at 4 different RIFLIP sites, and it matched perfectly. The chances that that occurred by chance are infinitesimally small. In fact, I think it's fewer than human beings that have ever existed on the planet. Here they are. These are 4 different RIFLIP sites, and you can see that's one in how many? One in a billion. The second site, 1 in 2 billion something. And when we look at different sites like this, you have to multiply them together to get the total probability. You multiply them together. So you end up, if you only look at 4 sites like this, you end up with 1 in trillions. That's the chance that it occurred purely by chance, this association. Very powerful to stand in front of a court and give them that kind of evidence with that kind of statistical backing. So the need for a lot of DNA limited the use of RIFLIP to those cases where there was a lot of semen or saliva or blood deposited at a scene, but it does not work for the trace amounts of DNA. You just don't end up with any results In addition, it's quite a tedious process, and it has been superseded by a much more rapid and in many ways a much easier method to do, but it is based on the same kind of principles. But here we must also go back to what we saw about the replication of DNA, because it uses the mechanics of DNA replication to actually artificially increase the amount of DNA that we are dealing with. So it is the technique we're going to describe now is very useful because you can take extremely small amounts of DNA. You can take DNA from just a few cells and you can amplify it. You can take a small amount of DNA and amplify it to a large enough amount of DNA that you can actually analyze. And this makes use of a laboratory technique called the polymerase chain reaction, or PCR. And many of you will now have heard about PCR because I'm sure many of you have had a PCR virus test, and you'll understand after this exactly how that test worked. So polymerase chain reaction is a technique that we use for taking very small amounts of DNA. You can even work with DNA which has been degraded into small sections. And you can, by running this polymerase chain reaction, we can increase the amount of DNA. What we're going to do is we're going to use the same replication mechanisms that the cell used to double up its DNA. We're just going to do it repeatedly. We're going to do it over and over and over. That's why it's a chain reaction. So remember that when we talked about the replication of DNA, this is what happened. We split the DNA strands. This is a DNA molecule, and it has sequence of bases. And on the other strand here has exactly matching sequence of bases. Where there is a T here, there would be an A here. Where there's a G here, there'd be a C there. We separate these two strands apart from one another. And in normal replication, the first thing that happens is a little patch of RNA, a primer, binds to the DNA right there, and it binds to the DNA right there. And it is matching. If this is an A, then that's a T. If this is a G, then that is a C. It obeys the same rules. It obeys the same base pairing rules. It's just that it's very short. They're usually no more than 15 to 30 bases long. They've made them look even shorter here, but it's not very big. Here's a really important point. This for us is key because there's a couple of things to notice. First of all, when replication occurs, when we double up this DNA, it's only going to occur here. It's going to start there where this was bound, and then it's going to carry on there. It's going to start there, and it's going to carry on there and replicate. But this here is binding to a specific place in the DNA. Okay, it's binding to a specific place. This means if I can make these primers, if I can manufacture them and put them in, I can start DNA replication at specific places in the molecule. I don't have to replicate the entire molecule. I only have to replicate a smaller area of it, and it starts right where I want it to start. What I have to do is I have to know that sequence. I have to know that DNA sequence. As long as I know that DNA sequence, and I know that DNA sequence there, I can make primers that when I put them into solution with the DNA, they will stick there and they will stick there. And replication will only occur from there on and from there on. And replication, by the way, will run for several thousand bases. It doesn't run forever. It runs for a while and then it will stop. And what I'll end up with are fragments. I'll end up with fragments there and there. So this is exactly what we do in PCR. First of all, we're going to take our DNA. Here's our DNA molecule. Here's the bases matching one another. And what we're going to do is we're going to make a solution. We take the DNA, we put it into solution with lots of DNA, with the primers that are going to stick there to the DNA. Then we're going to put in a special enzyme called DNA polymerase, and that is the DNA enzyme. That's the enzyme which will replicate the DNA. As long as the primer down here, the DNA polymerase can start working and do the replication either way. Then I'm going to put in lots and lots of nucleotides because we're going to have to we're making new DNA. So we're going to have to supply plenty of nucleotides to actually do that. So here's how this is done in a machine. It's called a thermocycler. And it's a thermocycler because what's going to do is going to heat up and cool down, heat up and cool down. This molecule of DNA, it's very easy to separate those strands. All I have to do is heat it up a little bit. So I'm going to heat this up, and I'm going to heat it up quite hot. I'm going to heat it to 95 degrees Celsius. It's 5 degrees away from boiling. What happens at that temperature is that the DNA strands actually separate from one another. Now, this enzyme here, the DNA polymerase here, is resistant to heat, so it's not killed by that high heat. Now I'm going to drop the temperature a little bit, and now at this lower temperature, these primers are able to bind. It's still too hot for the DNA fragments to get together completely, but it's cool enough that these little short pieces of RNA, of primer here, can actually stick to the DNA. Now I'm going to reduce the temperature even more. I'm going to reduce it to 72 degrees Celsius from the 95. And at this temperature, this polymerase is going to start operating, and it's going to actually replicate the DNA. So at the end of that step, we've gone 95 degrees, 55 degrees, then we've raised it again, 72 degrees, and we have doubled up our DNA. We've now got 2 DNA molecules where we had 1 before. Okay, that's great. Now what do? We do this again. We now push this back up here. We push the temperature back up to 95 degrees. These 2 strands separate into 4. We drop the temperature to 55. Primers stick to them. We raise it to 72. DNA polymerase doubles up the molecules, and what do we end up with? We end up with 8 molecules. Then we do it again, and we end up with 16, 32, 64. We can repeat, we usually repeat the cycles anywhere between 28 and 35 times, by which time at the end we have billions of molecules of DNA. Just these segments here, whatever has replicated here, and we've repeated it over and over again, and we have amplified from a few DNA molecules. We can amplify to abundant DNA molecules, which we can then type. So this is exactly what is going to be done. This is the polymerase chain reaction. When you had a polymerase chain reaction test for COVID, what they did was they had primers which sat on viral DNA, and it amplified that DNA to show whether or not viral DNA was present. If it was present, you're positive for COVID. If there's no amplification, you're negative. That's how it works. Okay, so the advantage of this polymerase chain reaction is that we can actually make use of another kind of tandem repeat which occurs in the human genome. It's very common in the human genome. There are many sites at which short tandem repeats occur. Remember that the RIFLIP depended on those very big repeating segments. Short tandem repeats are very short. They're often 4 or 5 bases long and repeating over and over and over again. And there are many sites that have been identified in the human genome, and the flanking DNA has been sequenced. So we know what primers to design. We can design primers that will sit on either side of those repeat areas. And in this case, we don't need much DNA at all. We only need a very small amount of DNA. And because we're using polymerase chain reaction, we can amplify and actually examine for the presence of the repeating segments. And we design it so that it's just the repeat segments that are replicated. So if I have short area of repeating segments and I run PCR, I'm going to end up with abundant short fragments. If the area is longer, I'm going to end up with abundant longer fragments. And I can separate them out. Again, using a technique, any technique which will cause the molecules to separate out from one another in such a way that I can examine them for them. You can do it by gel electrophoresis. It's just gel electrophoresis at the end is rather tedious to do for individual samples when you've got lots and lots of samples. Instead, we have automated mechanisms now, which depend on using capillary tubes to essentially do the same thing. It separates out large and small molecules in order, and we can examine for them as they emerge. Okay, it's the same principle as we saw for RIFLIP. Okay, you inherit these short tandem repeat areas from both mother and from father. There's a huge amount of variability in the human population. So there is a particular statistical chance that you will end up with a particular inheritance of one number from your mother and one from your father. And there are numerous short tandem repeat sites at different places in the genome, each with their own unique sequence that they are repeating, each with their own unique flanking areas that we can design primers for. It's a very, very useful technique. So the other huge advantage we can work out the statistical chances of inheriting at a particular site. But as we look at more and more and more sites, we multiply those up until we end up with enormously significant probabilities of this being unique to a particular person. So here's what we're going to do. It's exactly the technique that I've described to you for polymerase chain reaction. Just imagine we have an area here of the tandem repeats. In this case, there's just 3 short tandem repeats, and we have flanking it. We have an area of sequence. In this case, it reads CCAG, and then on the other strand it reads GAAT. And we're going to make primers. We're going to make primers which stick here, and we're going to make primers which stick here. And we're then going to run it through polymerase chain reaction, and it's going to produce fragments. And it's going to produce fragments of a particular length, and the length is going to be proportional because we are going to be able to extract our fragments, our restriction fragments there, not restriction fragment, I'm sorry, the tandem repeat fragment there and there. And we're going to be able to run them out on a gel to see the size. And we do this in actual fact for up to 20 different sites. The standard set by the FBI now is that we look at 20 different STR sites in the human genome in order to establish identity. So the statistics just become enormous. The validity of the technique is very, very big. The short tandem repeats are really short. They use 3 to 7 bases. And often the actual fragment itself is also very, very short, and it's about 450 bases, something which is ideal, because in PCR there is a limit. The amount of replication that takes place is limited. It's not like in the cell where it runs for hundreds of thousands of bases. In PCR, it's relatively short, the replication area. There's another advantage to the fact that these tandem repeat areas are short, and that is very often identity can be extracted even from very old DNA, where the DNA, what happens to DNA when it ages, especially if it's in the light, is it breaks into fragments, it shatters into small fragments. Those small fragments may still be useful, however. They may be long enough that we can extract STR information from them. Okay, so let's have a look. We'll have a look at a real site, a real STR site, and this is one called TH01, and I always call it 41, but I don't know if anybody else does. So it's an area with short tandem repeats in it, and the repeat is AATG, just 4. And everybody has the 41 site. It's just that different people have a different number of repeats, and it's usually between 5 and 11. And you can inherit a different one from your mother and from your father. You could inherit the same. You could have your mother and your father both had the same number of repeats at that site. But we're going to extract 41 using polymerase chain reaction, and we're going to see what the length of these different sites is. So here, this is what they look like. We've got one, let's say the top one is from your mother, bottom one from the father. And this one here, they are AATG, 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 etc., etc. And the top one has 6, and I think the bottom one has 8 repeating segments. On either side here and here, sorry, there is a sequence which we know. And so we can design primers, and we can run this on PCR, and we can amplify these fragments. We'll just amplify that, and we'll amplify that there. We run the PCR, even if we've got very little DNA. It doesn't matter, because we're going to run the PCR over and over, and we're going to amplify, and we're going to end up with abundant DNA just of that length there, and abundant DNA of that length there. And the reason is we knew what primers to make. We knew to make a primer for there. We knew to make a primer for there, so that replication would only take place there and there. So when we now go and we actually look at, maybe we have a crime scene and we have a suspect, various suspects, their DNA. We're going to run one PCR, and we're going to amplify for these. Well, here, have a look here, and you can see here's the crime scene DNA there. Here again is, by the way, is the standard. These are our size markers. Here's the crime scene DNA, and these are all suspects. So we're looking here at the FO1 site, okay? This one here, both mother and father were exactly the same. They had the same number of repeat segments. This one here, the mother and father were different, that had one very small fragment, maybe 2 repeats or something like this, and then one larger fragment, maybe 8 or whatever. There's only one suspect who actually matches, and here it is here. This person at that particular site for the FO1 site, for one site had, when we ran the PCR, we produced 2 fragments, one there, matches, and one here, matches, exactly the same site. This is only for one site. Nonetheless, we can work out for the general human population, we can work out what this, in this case, they had 6 and 8 that they determined. What proportion of population has that combination at the FO1 site? Only 3.5% of the population carry 6 and 8 as their combination. Okay, only 3.5%. Well, that's not enough for individual, right? It's not enough for individual characterization, but we run this for many sites. There are many of these sites on the genome, and as I've told you, the FBI has identified 20 different sites. And when we look at the probabilities at each of those sites, we multiply them to one another, and we end up with an enormous probability. If we have a matching for various sites, we end up with a very high probability that DNA from a question sample is the same as DNA identified from a standard sample taken from a suspect. Okay, so I'll end that there for today. We'll finish this discussion on DNA at our class on Monday. Okay, and I believe you have questions that you want to ask.
Yeah, I just wanted to know about the final paper.
Final paper, yes. So what happens, you've had 3 class tests. So your final paper, there is another class test in the final paper. That's on stuff which has not been examined yet. And then there is another test that effectively that runs at the same time, which is comprehensive. It's stuff that they may be reworded, but it's material you have been examined on before. It's based on questions that you have already seen. So you need to go over your previous tests very carefully, make sure you understand where you went wrong or whatever. And then you need to study the material which has not yet been examined. And that's the whole final.
Okay, and also you sent an email.
Sorry, say that I didn't hear you.
I'm sorry, you sent an email about the final paper. At the
Yes.
same thing or different?
No. Your final paper is that your essay on a crime. It's that the essay on the crime, which has to, I believe, you have to submit by the end of the month.
Okay, got it. Thank you.
All right then. Have a good weekend.
You too.
Yep. Stay safe.